Alright, these are your notes over transcription. Our essential question: how is DNA copied in transcription? The first step in making proteins. Alright, this is your outline. First we're going to talk about what proteins are, a protein synthesis overview, and then we'll go into part 1. That's a protein right there. So proteins. Here's a protein. Of our 4 macromolecules, we have fats, carbs, nucleic acids, and then the protein, that was the doer of the cell. It was like your meat, your fish. Here's some protein powder if anyone wants to build muscle. And you take protein drinks, protein bar, and then that's our protein synthesis. That's how it happens in the cell. So that's what proteins are right there. Protein synthesis overview. Okay, so jot this down. We've got part 1 is called transcription. Part 2 is called translation. In part 1, we go to the DNA and we are writing down the message. You'll have to draw in the RNA polymerase because they don't have our first blobby. RNA polymerase, it makes the message or copy inside of the nucleus. And then that message goes out, and then it goes out to the ER where we have our ribosome sitting. That's our ribosome, it has 2 parts to it. And then, you could draw in a little tRNA, and then each tRNA has a dot on top of it. And then we will learn about that, and it transfers over, and then we have amino acids, and then our line of amino acids makes a protein. Why don't you just follow along and we'll get it. Alright, so part 1, transcription, occurs in the nucleus. You copy the instruction manual, or DNA, or that's our cookbook, to make the message, or mRNA. That's what that M stands for. That was like the one recipe that you needed to write down. And this is, transcription is the first of 2 parts in protein synthesis. Synthesis means making a protein. So this guy right here is my scribe, and he is scribing something down. So transcription is writing it down. Good. Alrighty, so these are our steps. First, you've got RNA polymerase, the big blobby right here. He is going to break and unzip the DNA. So we've got our DNA right here. He's going to break it open. And then number 2, mRNA is made by copying the DNA. So if it's a G, it's going to bring in a C. And then the message, bam, leaves the nucleus and heads over to the second part of protein synthesis. And DNA closes back up after the message leaves. It closes back up and is left unchanged. My DNA is super important and never changes. We just have to make this tiny little message about what we need, and then it leaves. Alright, the mRNA message is similar to DNA, except for 3 important things. 1, 2, 3. DNA has a T, thymine, and RNA has uracil. That's a U. So now we have AUCG. Okay. The deoxyribose sugar, our gray snowflake looking guy, is replaced by a ribose sugar. That's our yellow or orange guy. And that's where you get the D for DNA and the R for RNA. And number 3, DNA is double-stranded and RNA is single-stranded, so 2 and 1. And then this is a picture right here of RNA, DNA. Okay, I want you to practice. Don't look. If this was DNA, CCGT, blah, 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 transcribe it into mRNA. So write blah, 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 blah. Okay, you could pause it if you want to and practice that. Alright, last thing, I want you to quick check, label, and describe what is happening. So, here's one picture we've looked at. Here's the DNA you built. Label DNA, RNA polymerase, mRNA, the base pairing rule A, U, G, C. Be able to do that and go do your Quizlets and have a great rest of your day.